Guys, I'm not going to, I don't think I'll record all the sessions, I don't know. But again, I'll tell you, just if you didn't understand something, just go to my YouTube channel and then watch them there. But it's different because right now, if you don't understand something, you can tell me now and I can explain. Well, also on YouTube, you just write a comment and then I might not reply on the very same day. So just use the videos on YouTube just for revision. So by now you know that it's a phospholipid bilayer. You know we have a polar head and a fatty acid that is hydrophobic, right? And then you know that we have 3 proteins attached to the membrane. We have glycoproteins, we have peripheral proteins, and we have integral proteins, right? And then I said to you that glycoproteins have 3 main functions, and I'm pretty sure that they will ask you. Number 1, I said they determine your blood type. Number 2, they act as receptors. Number 3, they're responsible for what? Cell recognition. Right?
Yes.
That is what you have to know. And then I spoke about, you remember, you see this red structure in between the fatty acids. I said that is your what? Your cholesterol. And when we were doing the integumentary system, what did we say about the cholesterol when it gets the sunlight? What is it converted to? What is an active cholesterol molecule converted to when it gets sunlight? You forgot.
Is it not vitamin D3? Vitamin.
Don't say is it not? Say it. Vitamin D. Yeah, you're correct. And then this cholesterol in between the fatty acids, what it does is that it stabilizes the membrane and it keeps it flexible. They'll ask you this one again. I can assure you. I'll repeat. The cholesterol molecule in between those fatty acids, its function is to stabilize the membrane and to make it flexible. I won't write that down. I hope you got it.
No, repeat, please.
It stabilizes the molecule, the membrane, and it makes it flexible.
Okay.
You're sorted.
Yes.
Then, before I can even forget, with your glycoproteins, glyco is you think glucose, right? So it's your sugar and your proteins. So we know that sugar is very sticky, right? Have you ever maybe slept with sweet in your mouth or something? Then for some reason, or just hold the sweet, right? Just hold it. And then after a few minutes, you'll realize that your fingers will be sticky. Do you know what I'm talking about this?
No.
You don't know that sugar is sticky?
We know that.
Okay, thank you. And then forget what I just told you. Just know that sugar is sticky. I was trying to explain that. So since sugar is sticky and we are saying that this is sugar plus proteins, these glycoproteins, they form some sort of a glue on top of the membrane, right? And what do we call that glue? We call it glycocalyx.
Okay.
That is what you have to know. Are we on the same page?
Yes.
So that is it for the cell membrane. That is what you have to know about the structure of the cell membrane. And
What
then
is the function of that glycocalyx?
it can be used as a receptor itself, but not most of the time. Mostly it's used to trap foreign particles.
Particles.
Yeah.
Okay.
Are we fine, guys?
Yeah.
Oh, sure. And then going to let me quickly wipe everything quickly. So we don't have a busy screen. I want us to talk about junctions right now, because we just spoke about the cell membrane and we find our junctions in the cell membrane. So let's see. So what you have to know, guys, is that we have 3 types of junctions. I'll show you a picture of junctions when I'm done explaining. So Nobuhle, your hand is raised. How do large substances pass the membrane? Thank you for answering. For Jesus, I almost said answering that question. Thank you for asking. I don't know why I didn't even explain that. Jesus. Your integral proteins can be used as channels, right? So some molecules can pass through, right? And there are other proteins besides the integral proteins. There are other proteins that form a passage right here. And we are going to talk about this when you're I think we should do cell physiology separate, right? To talk about transport mechanisms. That is when you will understand how larger molecules pass through. Because right now I feel like I'll be confusing you and stuff. I just want us to have just one session, one separate session, whereby we'll just be speaking about the active processes, the passive ones, diffusion and filtration, and all those things. Like how do substances move from the outer cell into the outside, from the extracellular fluid to the intracellular fluid. Are we clear? Because if I'm going to explain that right now, I'll waste time, guys. And I said I just want to push so that you have enough time to prepare for biology. So they have a lot of information. So hence I want to just do a separate lesson. So the 3 junctions that I want you to know, the first one is tight junctions, right? I'll just write TJ, tight junctions. So your tight junctions are also called impermeable junctions. Your tight junctions are also called your impermeable junctions, right? So what do your tight junctions look like? A zip, right? So if this is your cell, if we have cell number 1 here, and then we have cell number 2 here, this obviously in green, that's your cell membrane, because you only find your junctions between the cell membrane of one cell and another. So your tight junctions would be here, like this, holding these membranes together like that. So they look like a zip. That is what they use sometimes when they ask you questions in your exam. The moment they say a junction like that, it's like a zip, you should know they're talking about tight junctions, right? So what is the function of the tight junctions? They are very impermeable. They don't let stuff to pass through, right? They don't let any stuff to pass through between the cells. So they hold the cells together. Are we clear? It will be easier for you to remember it because the word tight just gives you a clue. Tight junctions, they hold the cells tightly. And anything that is tight, obviously, it's impermeable. That is all that you have to know about tight junctions. Don't confuse yourself. Secondly, the second type of junctions that I want you to know is your desmosomes. We spoke about desmosomes when we were doing the tegmental system. In which layer of the epidermis did we say we find the desmosomes? Guys, did you forget?
No.
Talk to me then.
What are you saying?
In which layer of the epidermis do we find the
Yeah.
desmosomes?
Stratum spinosum. Oh,
The spine.
spinosum.
I'm telling you, people are ready. You're ready for the exam, eh? So your desmosomes are also called anchoring junctions, right? So they anchor. Because remember when we were doing the integumental system, I said they hold those spiny cells together. They hold them together. So what do? They prevent cells which are subjected to mechanical stress from being pulled apart, right? Which cells are subjected to mechanical stress? Like your skin, for example, right? Your skin can there's friction day in and day out between the surfaces, maybe just like you're sitting right now in your chair or whatever. You're rubbing your skin against that chair or against that table and everything. So there's friction. So the desmosomes prevent those skills from the skin cells from moving away from each other. So they anchor the skin cells, right? They prevent cells that are subjected to mechanical stress from being pulled apart. And then you have, if this is one cell and this is another, you have something we call a plaque, right? Cell will have a plaque here. This will also have something like a plaque, and then you will have your communication between the two, right? This, the whole of this here, the whole of this, that's your desmosome. What we have in between here, we call it cadherins. Because people tend to think that cadherins only, they say that's desmosome. The whole of this is a desmosome, not this in between. And then this is your plaque. And then you will have filaments going from your plaque to the inside of your cell. You will have filaments here, like that. You are going to understand this more in biology because they go into deeper detail, right? But this is what you have to know about desmosomes, is that they have thick protein filaments which extend from one plug to the next. Which are those proteins? Catenaries. Are we clear? And then the third type of junctions that I want you to know is your gap junctions. You just write gap chain. So gap junctions. Your gap junctions are also known as your communicating junctions. So they can either say impermeable junctions or tight junctions. They can either say anchoring junctions or desmosomes. You have to know these words. They can say gap junctions or what? Communicating junctions. I won't write communicating. Okay, let me write communicating. These are your communicating junctions, right? So how do cells communicate using gap junctions? If this is your cell number 1, this is your cell number 2, you will have something that will be like that, right? In between, like this. So what do we call this? It's your gap junctions. So inside your gap junctions, obviously there's a hollow tube. It's like there's a tube coming from inside this thing. What do we call that? Hollow tube, we call it a what? A connect zone. The only way, guys, you'll remember all of these words is if you study. If you don't repeat, so trust me, you'll only remember them for today. So this is your connect zone, and your connect zone, as I just said, it's your hollow cylinders, right? They are water filled. They're water filled. So cells can communicate using connect zones. Are we clear? And your cap junctions are your transmembrane proteins. You can also say transmembrane proteins. You can say communicating junctions. So you have to know. So far, sorry? Do you guys understand?
Kevin, can you please redo the gap junction? Can you please redo the gap junction?
Okay, listen to me. I'm saying gap junctions are also called communicating junctions. That's the first thing that you have to know. And your gap junctions are transmembrane proteins. You want me to write that down or record it?
I got it.
Okay. And what you have to understand is that in between those gap junctions, we have a tube-like structure, right? Which we call it what? A connect zone. So I'm saying that your connect zone is what? It's your hollow cylinder, which is water filled, which helps one substance to move from one cell to the other. That is all you have to know about gap junctions. Are you clear? Didn't understand me somewhere? What do you need me to clarify?
Okay, thanks.
You do understand?
Yeah,
Sure.
yes, I'm sure.
All right, if you say so. Others, do you understand? Because I'm about to wipe everything. Okay, teammates, your hand is raised. Do you have a question? Okay, teammates, do you have a question? No, I don't press it by mistake. Why do you always press the hand by mistake? All of you, since we started with these sessions, you've been pressing the hands by mistake, or you're scared to ask. So I just want to show you this. This picture is, so you see how your tight junctions look like. So this is just a cell, they cut it in half. This is cell number 1, cell number 2, and this is cell number 3. And this is your membrane. You can see that it's a bilipid, phospholipid, right? It's a phospholipid bilayer, not bilipid. What am I saying? See? So these are your tight junctions. So they have what? They have connexons. Don't mind this one. Don't mind the adherence. And your hemidesmosomes, don't mind them. You focus on these ones when you're doing biology. Okay? Don't really mind them for now. Gap junctions, these are the gap junctions. You can see the gap. So the gap junctions are the ones which have connexons, right? The tight junctions are like a zip, you see, like here. And then your desmosomes, these are the plugs that I told you about. And these in between are your cadherents. These are the filaments. Are you guys clear? Because I'm deleting this picture. Are we good? So we spoke about the nucleus. I told you what the nucleus is, because I said your cell has the nucleus, it is a cytoplasm, and it is a cell membrane. I just spoke about the cell membrane. I told you the phospholipid bilayer. I told you the proteins that we find there and everything. I told you about the nucleus. I told you about the nucleolus, the nuclear membrane, and that the nucleolus is where your ribosomes are synthesized. You understand that? Now we go to the cytoplasm. What I want you to understand about the cytoplasm is I told you that it has 3 things. It is your cytosol, your inclusions, and your organelles. And I said your cytosol is a semi-transparent fluid, right? I said it is largely water. I said it is largely water and it has some of the nutrients, right? And then your inclusions, I said it's your stored nutrients or products that are floating in the protosol, in the cytosol, sorry, right? And in addition to that, I said, if you ever get confused with what's an inclusion and what's not, just know that your inclusions are not membrane bound, right? And then your organelles, I said it's your little organs which are specialized to carry out specific functions. And for the mere fact that your organelles have their own membranes, right? It's because of something we call compartmentalization. I don't know if you want me to write it down, but compartment, it's that it has its own the organelles have their own membranes, right? Compartmentalization just simply means that the organelles have their own membranes. So their own membranes help them to have their separate environment with whereby they can manufacture wherever they have to manufacture and then take the products into the cell or outwards of the cell, right? So from now on, we are going to do organelles. We have many organelles in the cell, right? We're going to do organelles. So meaning that you all know cell, right? Meaning that you're going to look, we've already looked at the chromatin. You understand what the chromatin is. Nucleus, you understand. Nucleus, you understand. This is your nucleoplasm. And I just want you to get a clear picture of everything before we can even start, right? Your cytoplasm. So what we are going to do is, okay, guys, I won't write down things, right? So listen to me, tell me if you don't understand something, right? I'll explain while you're looking at this picture because I think it's easier that way. That is how we can be fast. Because if I can try writing things down, it will take forever. So we are going to start with the mitochondria. Here. What you have to understand and what you have to know about mitochondria, firstly, you have to know that it is the powerhouse of the cell. Because that is what they like. This, they've been telling me this since from high school. It's the powerhouse, right? Why do they call it the powerhouse? Because this is where we get our energy.
Can you please fix your mic?
My mic is fine.
No, Wakata.
Steve. Kiakata for everyone or just for you? Others, Kiakata.
Fine now. It's fine now.
Okay. So I'm saying that the first that you have to know in mitochondria, right? You have to know that your mitochondria is your powerhouse. And then your mitochondria manufactures what? Your ATP molecules. And that is all you have to know about the mitochondria. When it comes to the diagram itself, just know that you might. Okay, guys, we are left with 10 minutes. Do you want to come back after this session, or you want to take a break? We'll continue later or tomorrow. Or you want us to come back and finish with the organelles? Okay. What is saying we must come back? What are others saying? Let's finish with the organelles. If we finish with the organelles, that means we're coming back, right? What are others saying? Bali, what are you saying?
No, nothing. I'm just thinking out loud.
Do you want us to come back and finish with the organelles, or just this has to be the last lesson?
Yes, let's finish the organelles.
Okay. That's quiet, so I assume you agree with others to say that we come back for organelles. So that is all you have to know about mitochondria. In terms of its, if we were to zoom in, because that's your mitochondria, it is double membrane, right? There's the outer membrane, and then the inner membrane is not really smooth, like it has these projections like that, right? That's your inner membrane. And inner membrane, these projections, we call them crystal. And then it has the matrix and then it has granules. Don't say it has a cytoplasm. It doesn't have a cytoplasm. This clear structure, we call it the matrix. That's all you have to know about the mitochondria. As for okay, let me not do that right now because I confuse you. It's fine, forget it. And then the next organelle that you're going to focus on is your ribosomes. Ribosomes are not sure. These are your ribosomes. What am I doing? Do you see this rough endoplasmic reticulum? DC. What is attached to it? Those little dots. Those are ribosomes, right? So what do you know about ribosomes so far? I said that your ribosomes are manufactured from. They are manufactured from your nucleolus. They will ask you that question. Each and everything that I'm telling you, if I'm saying they'll ask you this question, I'm telling you, if there's no way you'll write all the tests and even finish with the exams without even getting one of the questions I'm telling you that they'll ask you. Ribosomes are manufactured in your nucleolus.
Okay.
And they are made up of proteins, and they have ribosomal DNA. Not DNA, sorry, ribosomal RNA.
Yes.
Right? Going back to your mitochondria, we have mitochondrial RNA. And everyone has their mother's mitochondrial RNA. There's no father's mitochondrial RNA. Do you understand why? Do you understand why, guys? We only have mother's mitochondrial RNA.
Why?
You are saying yeah or why?
Why?
Okay. During reproduction, when your when the sperm of a male comes and joins the ovary already has what? Mitochondria in it because it's a cell, right? So the only thing that this sperm, the sperm doesn't, not the whole of this cell goes into the ovary, right? Do you understand me?
Yes.
Not the whole cell. So it means that the mitochondria of the father doesn't go into the cell. And after this sperm releases its genetic material into the ovary, it's no longer important. It's useless. So meaning that the ovary now has only the genetic material of the father and that of the mother, but it has its own mitochondria. So that is why the mitochondrial DNA, mitochondrial RNA, it's maintained for generations. There's no father's mitochondria because the mitochondria doesn't even leave the sperm. I hope that makes sense. It's clear or I'm confusing you? The only thing that leaves the sperm to go to the ovary is what? Yeah, the genetic material only. The rest, they stay behind.
Okay. Mm.
So meaning that they can trace, if you are your mother's child, they can do it with mitochondrial RNA. But they can't say they'll use mitochondrial RNA to see if you and your father, there's no father in mitochondrial RNA. So that is why they always use DNA, because DNA is both your mother and your father. Mitochondrial RNA is only your mother. Are we clear? I hope we are. Thank you. So that is what you have to know about ribosomes, where they have manufactured nucleolus, and that you have ribosomal RNA, and that the ribosomes, they serve as the actual site of protein synthesis. Again, we do protein synthesis when we do cell physiology, which I think that one also must. So when we are doing cell physiology, we'll do the transport mechanisms and the protein synthesis and cell division, all of those, before we can even move to tissues. Are we clear?
Yes.
The next, since we spoke about the ribosomes, and ribosomes are attached to the endoplasmic reticulum, the next organelle that I want us to focus on is the endoplasmic reticulum, right? So your endoplasmic reticulum, okay, guys, we are left with less than a minute. So let me end the meeting automatically. Please come back, right? Because this, okay, let me leave it because it's left with seconds. So come back, that will be the last session. That will be the last 40 minutes of the day. Are we clear?